Good morning, everybody. Thanks, everyone, for flying in, even under shortish notification. Much appreciated. And so welcome to the Proposers Day for Delphi. For those of you who it's your first Proposers Day for an ARPA, today is for you. It's for the proposers. Oftentimes in an ARPA program, we're asking for people with diverse skills to come together, to team, to try to pull off something quite ambitious. Sometimes you may have, 80% of the solution and you're looking for that last 20%. In the Proposers Day and these teaming profiles is how we encourage you to do that, right? We put the capability out of it. So our first goal is for you guys to be talking. So on some level, apologize for breaking up the conversations. I hope you have more conversations later on over the posters. And then the second bit is to go through the program and as clearly as we can state exactly what we want out of the effort, right? So that way, as you write your proposals, you can know exactly what we're after and what to emphasize. There we go. Okay, so for today, the agenda, we already registered. You'll have my opening remarks about the effort. Then I'm going to dive in a little bit onto how ARPA-H works, for those of you who have not worked with an ARPA before. And then we have Adria Robinson, who's going to talk about acquisitions. Then we have some partners. Ruzba Jafari from DARPA is going to talk about the VITAL program, another way to sort of leverage some complementary capabilities that he is trying to develop in his effort. We'll have a short break. We'll set up some posters. This is going to be a great opportunity for you guys to learn from each other. After the poster session, we'll have lunch. During the poster session, at the beginning of lunch, that is when, if you have questions for us, you saw those cards that were handed out, right? Fill out exactly what your questions are. We will come up with a broad answer to it that tries to answer your question, but it may be other people. So if you have something very specific to your technology, we may sort of more generalize the response instead of a direct comment on your technology. We'll have that response to submitted questions. We're going to be have an efficient day. We'll close at about 2:30. Afterwards, we're going to have government team availability. If you want to have a bit of a sidebar, we're going to grab a portion of the room. Did you want to talk a little bit about the process, Ed?
So we're printing a sign-up sheet for sidebars. It'll be a 7-minute sidebar, but a minute of that is walking in and out of the room. We're going to really be strict on time. That'll be held in the Campanile room. So we'll have someone posted outside there. You're going to come in, come out. We can't say, hey, we love this technology, we don't love this technology. The best use of your time in that window is this in scope? Is this out of scope? Don't be trying to sell us on your product and things like that. Save that for the solution summary, save that for the proposal.
All right, perfect. Thank you, Ed. Okay, so a little bit about ARPA-H. I know some of you are old hands in ARPAs, but I just wanted to explain how we're organized, where we sit within HHS, and how we do business a little bit. So ARPA-H, of course, inspired by DARPA. It is a projects agency. We do short 3, 4, 5-year programs that try to achieve a capability in a very short period of time. And everything else that you'll see is really focused around that, enabling that quick transition to a capability. So it's an independent component of HHS. We're not part of NIH. We report directly to the secretary. That gives us a lot of independence to do what we think is best. We don't have our own internal research labs. There is no competition from us. We are strictly a funding agency. We hand out money to smart people such as yourselves to get things done. Roughly $2.5 billion initial appropriation. We're slated for another $1.5 billion this year. That enables us to aggressively fund programs. So the cost is always what the cost is always what is needed to get the job done. We generally fund contracts, not grants. I think many of you are familiar that ARPAs tend to fund generously. The downside, I always warn people, that means we're going to come and sleep on your couch, right? So we very tightly monitor what's going on. We're always in contact. Some of that's annoying, but the flip side is we're actually here to help. And people who have worked with us is that we're always making connections for you to other people, people that you should talk to, people that can enable you to get things done. And like in this program in particular, many people have come out, other organizations, this will come up, later, are interested in this capability, and they would love to work with performers who can produce some of the results that we're after in Delphi. We have other capabilities. We're able to directly reimburse FDA in order to, get quick eyes on technologies. All right. The health science, this is just a brief overview of ARPA-H. Health science futures, they tend to do the craziest of the craziest, expanding really what's technically possible. Scalable solutions is also pretty straightforward. Is that okay? Sometimes making one versus making 10,000 is a qualitative difference, the ability to massively scale. And there's a little bit of that within this program. Proactive health is how do we keep people from becoming patients? And then resilient systems, that's the current office that I am in, and that is how do we make the healthcare system more robust. The other interesting aspect of these ARPA projects is there's a bit of a flywheel. Everyone at ARPA-H is short-term, every single government employee, including the program managers. You come in, you're giving substantial resources to get things done, and then they kick you out, right? They don't want you to get comfortable. They don't want people to say, I've been here for 20 years. We tried it 15 years ago. It didn't work. Well, maybe it'll work now, right? So there's a constant influx of fresh blood. This is also where I say we need a constant influx of fresh blood. If you or any of your colleagues are interested in becoming program managers, let us know. We're always trying to bring new people in. Program formation, right, is we're sort of in the middle in this the challenge stage. A program manager comes up with some idea, something that might be possible, something that is difficult and that other people would find it challenging to fund. Screen's blinking in and out, right? The challenge should not be easily solvable through traditional activities. And that is one of the hardest things when they go around talking to people and they say, I have this amazing drug, I think it would do a lot of good. And I go, that's very important, but your approach just isn't an interesting approach, a new approach, right? You should be doing this, but there's not actually risk. You're going down a traditional pathway, and that is not how we do business. We want to go down a pathway or do a thing where you stay up at night a little bit worried that any of this is going to work out, right? So that high risk is a critical aspect of it. So we put out this challenge. Smart people come in, they have ideas. We go through source selection. We find the ones that we think will meet all the metrics of the program, and then we launch the program. And hooray! Everyone gets money. We kick off the program. Second half of that is this. We're very much a performance-based model. You will see the different metrics, right? And so throughout the program, there are go, no-go gates, where we're going to take a look to make sure the progress. And we're flexible. If someone put in, if we put in something that no longer makes sense, sometimes we can shift that around. It's like, you're right, that this no longer makes sense. This should be our new objective. But that overall goal of where we're trying to get is key, hitting those metrics to stay within the program. And that is important because sometimes the approach just isn't going to work out. You're smart people. You should be doing something that's going to work out instead of just trying to beat your head against the wall. At the same time, it also enables us to take those resources and focuses on the approaches that do work. So it's as we go through, we continue to fund the efforts that are working out well, and ultimately you wind up with success. The other aspect of this, I will bring back in the other thing. There's a bit of a flywheel. Sometime during this program, there will be a new PM, and that brings in fresh eyes, fresh goals for what the program should be. You'll still have the same metrics, but it'll be a reappraisal by somebody with a different skill set. All right, that's a little bit about ARPA-H. On to Delphi itself. So Delphi, get its name from the Temple of Apollo at Delphi, and as you walked in, what did it say? It said, know thyself, right? That's the beginning of wisdom. So what we're really after here is how do we give people significant self-knowledge of their own personal health? How can we have people enable people to monitor their health continuously? So we're after discrete, and that should be EET and low-cost wearable devices. We're after discrete and low-cost wearable devices that could help Americans continuously track deep biological information. And I'll talk about this deep biological information in a second. Actually, I'll just pop over, right? And so what do we mean by deep biological information? What I'm after are hormones, cytokines, other markers of inflammation, levels of drug within the body, right? And so there are many biosensors out there. What we're not after are biophysical markers. If you want to include biophysical markers that somehow improve the overall diagnostic, that's fine, but that is not the goal of the effort. Similarly, we're not after high concentration metabolites, right? So if you have another great glucose monitor, we're not interested, right? Or lactate. We're after hormones, inflammatory markers, drugs, lower concentration analytes. The other aspect that we're after is that we want to develop an ecosystem for developing new biosensors, right? So develop of wearable or ingestible sensors for basically in a biomarker. This ecosystem should have multiple components. These components should be modular. They should be inexpensive. They should be small and low burden on the patient, and they should be secure or communicate securely. If we can pull this off, there are many, many benefits, right? And this is sort of across ARPA-H agency. Almost everything that we want to do is based on having some knowledge of what's going on inside the body, right? We want early detection so that people don't become patients. We want continuous monitoring so you can move patients out of the hospital, right, with their lights and machines going bing and people running around to their homes wherever it's known that patients heal more quickly. And this also is going to enable precision therapy, right? If you want closed loop control over therapies, then you have to have high quality sensors. Otherwise, the rest of the closed loop falls down. So ultimately, we want to build the building blocks of biosensing. So the way we are going about this are through chiplets. Chiplets, as many of you know better than I do, are sort of where the industry is going. So where we are right now is in the world of the printed circuit boards. I've been funding things since 2017. I've paid for many, 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 many printed circuit boards. And this is done well. People are quite good at it. And on a basic level, I'm not knocking the high quality engineering that has been done. But what normally happens is you optimize this printed circuit board, and then with time and with a lot of money, you wind up with an ASIC, right? And again, if you have one specific application, that is fine, but that tends to be high cost and not generating enough numbers to really bring down the cost of this capability, right? And so for those of you on more the bioanalytical side, well, a chiplet is simply whether you take one of these ASICs, these systems on your chip, you break it up into your component capabilities, memory, I/O, power management, right? And then you package it all into a new substrate, right? And this enables you to bring down cost. I won't go into all the reasons the electronics industry is doing it, but it brings down your cost. It gets you better yield, allows you to heterogeneously integrate. Right. And I think this is going to be important for biosensors because silicon isn't always the best substrate for biosensing. So it gives you some flexibility in the substrates that you want, and then you can heterogeneously integrate them to a capability that's scalable, it's modular, it's ultimately more reliable. You can get improved power efficiency compared to printed circuit boards. And then there's flexibility in sourcing, right? So one of them, currently, if you're making ASIC, it all has to clearly be run out of one fab, but people have already shown that you can integrate chiplets from different vendors. And so, so where I think we should be is, over to these bio chiplets, using this chiplet capability to generate improved sensors, right? So what needs to be done to get to the to these bio chiplets? In ARPA programs, we have technical areas, TA1s, and really there are many other things that will need to be done. We feel that these are the most challenging aspects. of the effort, right? So technical area one are what we'll call dry chiplets. These are chiplets that are unique to biosensing. So in my experience, as we build out these, there are a few things that are a little bit different. One is power management. Many biosensors compared to a lot of electronics are off most of the time simply because electronics run much faster than the human body. And so the off state becomes very critical. On top of that, there's often a variety of funding of power strategies from harvesting RF to batteries. And so the ability to sip that power from these different sources is critical. Highly secure comms, right? And so in particular, I am thinking about post-quantum secure. I don't know what the most recent, what are they calling it, Q-day, like when they think a quantum computer can start cracking basically all of our passwords. I think the most recent guess is like 2029 within the time frame of this program. But the point is, if we're walking around broadcasting biomedical information, it needs to be as secure as possible, right? So highly secure comms. The second aspect are what we're calling wet biosensor chiplets. This is the one that interfaces with the biology, the chiplets that enable you to detect these low concentration hormones, cytokines, and drugs. Screen keeps popping in and out. There are many different capabilities that we need in here. Now, note if you can do this without directly touching the biology, and you can prove to us that you can do it from standoff technique, we would love to hear about that. But for many of the applications that I've seen, you need some interface with the biology. And I do think this is going to be one of the most challenging science aspects of this. People have published on being able to do this, but it's really sort of at the edge of what's capable. The third is biocompatible encapsulation, right? So we want packaging and encapsulation that is highly scalable, that is biocompatible, and does this trick where you are sealing off what we're calling the dry chiplets while also exposing the wet chiplets to the biology, right? So a bit of a challenge. And the last task area are, depending on your track, and I'll explain this in a minute, either human factors testing or a clinical trial. If we can pull off all of these different aspects, we can go from today, these large, bulky sensors, to the future with DELPHI are small, very discrete, barely noticeable biosensors that can track the body, hormones, cytokines, other analytes. So just diving a little bit into each one of the areas. So for the dry chiplets, as I was mentioning just a moment ago, the requirement for many of the biosensors is that the electronics can differ substantially from more conventional electronics. Power is at a premium, especially if something is ingestible. They're often in the off state, right? And so being able to sip that power and generate highly secure communications is critical. You see the metrics there. These are the same ones from the solicitation. But here it's just different hitting NIST security standards for the most part, and the ability to operate for an extended period of time, 1 week. Lots of enabling technologies here. We're really just talking about the sort of the diversity of power strategies that people have come up with, from harvesting power. And look, a battery is fine if you can use a very small battery. There are other strategies that are out there, different ways to harness the RF that is out there, different ways to harness the movement of the body, but a battery is also fine. There have also been reports, I'll let you do your own Googling, on low power security protocols. This is broadly of interest, and here there's a need to bring that into a chiplet format and integrate it with your electronics. TA2 is the wet biosensing chiplets, right? So this is, again, one of the more challenging aspects of the program. Currently, most continuous biosensing is either biophysical, right? Heart rate variability, I would even throw pulse ox and things like that into it, or it's high concentration metabolites. We're really trying to push the ability to detect other soluble factors within the body, the way the body communicates with itself, right? So these are the hormones, these are the different cytokines. And part of the goal in your proposal is talking about what you want to track and why what you're tracking is important, right? And how you're going to provide high quality information. So in this TA, we're going to focus on the sort of the biology facing sensors. for detection of real or deep biological information. There's a bonus for anyone who helps me come up with a better word for what I'm after. I think we all get it when I say the deep biological information. And here, as we step through, you see within 12 months, you need a sensor that has a limit of detection that's at least below 10-fold. What is the physiologically relevant level, right? And so again, this is something you need to have a number for. If you are tracking, hormone X, what is the concentration of hormone in X, right? And how are you going to reach that low of concentration? And then as we step through, we're not making just one sensor. We're going to be making multiple sensors within this program. This is part of what I was saying earlier, that I want an ecosystem that is flexible, enables you to generate multiple sensors. All right, so lots of enabling technologies. Again, you can go out. There are quite many papers out there on the advanced design of molecular probes that can bind with high sensitivity, high selectivity, but that also enable continuous monitoring of these analytes. Robust detection probes, ones that can work in these biological fluids, have also been worked out. Here's one listed. Aptamers is one solution, is not the only solution. Other people have designed it. We are open to novel approaches for detection, right? So that is on you to propose and justify to us. Even beyond that, we're open to other capabilities here that will improve the performance of the sensors. What one finds often when you're building out biosensors is that you start off thinking it's the transduction mechanism, and then you spend years on optimizing the surface chemistry. So we're open to new ways of optimizing surface chemistry. I will throw out, is not a requirement, but for instance, there are self-driving labs, AI-driven labs that do a great job in optimizing the performance of enzymes or the performance of different molecules, proteins, right? So one might think of using an AI-driven lab to optimize the surface chemistry. But even beyond that, we're also open to other ideas of the ability to perform this post-processing, this surface chemistry, basically adjacent to the lab. If there could be a separate hood, a separate bench, these sensors, come out of the fab, they get quickly packaged, and then you can do the chemistry. For those of us who have built sensors, they know the fresher the surface, and normally the better the chemistry. So we're open to new thoughts on how to improve the performance of the chemistry. The last is the biocompatible encapsulation, right? And so especially for sensors that require access to biological fluid, you have this dual challenge. You need to seal up most of the electronics, right? And I will say, I remember as a postdoc working in bioelectronics and then having quite a few fabricated devices explode when exposed to buffer, right? Because the sealing of the sensors are off, right? You have to keep the dry sensors dry, at the same time expose the wet sensors to the biology. So we're open to new concepts there. You see the mil specs, the ISO standards. There are quite a few. Part of this is the packaging, pulling together the different components that has been automated. I'm not sure too much innovation has to happen there, but open to new capabilities or best implementation of best practices. There are lots of other chemistries out there. People do ALD, people have developed Parylene C, there are hydrogels, there are other encapsulation technologies. Doesn't have to be as aggressive, of course, as for implantable sensors, but we just need to make sure that you have a sealed device, right? Ultimately here, this TA3 is working towards an end-to-end vendor-neutral chiplet assembly capability. All right, so those are the three more technical TAs. TA4 is, of course, critical. ARPA-H is highly translational. We want to get something to the patient, right? So to do this, we need to achieve scale, and we have to think how we're going to address the regulatory framework. So if you look at the scale, right, if we really want to have high performance, low cost biosensors, we need to scale, right? Scale not just tens, hundreds of patients, but to address millions of people, tens of millions of devices. So to get there, it's going to be imperative to minimize regulatory burden. The FDA earlier this year put out some very interesting guidance along those lines, right? So just to step through, if we think about, first non-invasive, right, doesn't break the stratum corneum. If you think of minimally invasive or ingestible, and we're just on the left side of the screen here, or implantable, as you go from non-invasive to implantable, the scalability tends to go down. Fewer and fewer people want something that's an implant. At the same time, as you go from non-invasive to implantable, the regulatory burden goes up. Now, why do you want to move towards the right? Well, there's more biological information when you're in vivo or implantable. Our judgment is that not moving all the way to implantable is the best way to go if we're going to hit the scale and generate the insights for the biology. And this is really where the challenge is. How do we stay non-invasive, minimally invasive, so that we can scale and still achieve this biological information? So going back to going to the non-invasive, we feel that this is going to drive the scaling and faster market adoption. Again, this is a device that does not break the stratum corneum. Examples are, wearable estrogen sensors. If you can pull off, photoacoustic interrogation of the blood and pull off, again, tracking analytes that way, those would be examples. If you can do this, and if you're not giving clinical information, and we're okay with your careful wording on this thing, the FDA says that if you're promoting, tracking, or encouraging choices that is part of a healthy lifestyle, may help to reduce the risk of certain chronic diseases or help living well where certain chronic diseases or conditions, right? You don't need to go through the FDA. Right. And so that is a very easy way to reduce that regulatory burden and be able to scale quite quickly. So in that case, your phase 3 is not a clinical trial. It is a human factors testing, right? And then all the way on the right-hand side, getting to minimally invasive, we do believe that if you break the stratum corneum or ingest it, it's likely that you're going to get a greater number of biomarkers. It's going to lead to the broadest number of uses. Wearable CGMs are already a good example of this, ingestible GI tract monitors. In this case, phase 3 is going to be a clinical trial, right? And that's okay. Well, now the cool thing, at least according to DELPHI, is like, a device that is non-invasive sitting on top of the skin and a device that is, minimally invasive from the electronic standpoint is the exact same device, right? There's very little difference. And so we're enabled to extract this deeper biological information from minimally invasive, and we're able to scale to the non-invasive. So we're looking for both approaches. And happy if people are asking about that balance, you can put that in the questions, and we're happy to go back and forth on that. As noted, TA4, human factors testing for non-invasive, minimally invasive. Scalability is important in this program, right? And so I just wanted to highlight this. We also bolded within the solicitation. Proposing teams must have a strong and demonstrated capability in scalable manufacturing commercialization. If you say, we're going to do all this, and at the end we'll buy a fab or something and start production, right? We're not interested. You actually have to be in the business of producing biosensors and microelectronics, right? So please make sure that you partner, find some way or to convince us that you can actually scale these capabilities. And this will be bolded in the solicitation. So the program structure, in some ways, it's a fairly conventional structure where in phase 1, we're going to build up the components. Phase 2, we're going to integrate them, preclinical, begin to test them. And in phase 3, it's either this human factors or a clinical trial. The one thing that we're doing differently is that we want to be able to do a remix, right? What we're calling remix, where we take the components that have been developed and develop a new sensor, right? And so you, for phase 1, you're going to come to us with the biosensor that you want to develop. We're going to also ask you for a short list of other things that you could potentially detect with your technology. So phase 1, you're going to be going after that first one. Phase 2, we're going to pick from your short list something else that you're going to go after. Phase 3 is going to be a wild card. as noted, there are many other organizations that we will be able to talk about after source selection that are interested in using this technology, but who want biosensors for a variety of applications. So there's even beyond ARPA-H, there's a lot of funding available for this. So that's going to be an opportunity to go after a broad range of factors, but you don't know that up front. And this is one of the reasons why an ASIC probably is not going to cut it, because we're going to may have something completely different needs. And so the ability to have a modular remixable system is going to be critical. So again, phase one, you're going to go after, what you propose. Phase two, we're going to select from your short list. And then phase three, you're just going to be told, hey, here's something brand new that you should be going after. You're able to work with other teams. Maybe they have a component, a chiplet that you could use, you can remix, and we will work that out within the program. Ed's going to make an announcement. All right.
Well, a lot of the people online recognize there's no ability for you to ask a question. If you have a question for us, submit it to delphi@arpah.gov. That's delphi@arpah.gov. We'll include that in the questions, the cards that we're looking at during lunch, and Paul will address those questions, as many as we can. At the end of the day, sorry, we don't have an ability for you to ask questions directly or interact with us. We appreciate your ability to make this meeting quickly. We also had to assemble this all quickly. So and with that, Adria.
Good morning. I'm Adria Robinson. I am the ARPA-H Agreements Officer who will be supporting Paul and his team on the DELPHI program. All right, quick summary. So Paul mentioned that at ARPA-H, we don't award grants. Those of you who have worked with NIH and other government entities on research and development in the past are probably used to grants and cooperative agreements. Paul used the word contract. We use that as a little C contract, not a big C contract, right? So those of you all who might have received contracts in the past from government agencies, we use other transaction agreements, and I'll talk in a moment about what those look like specifically. In the ISO, what you've seen in the draft and what you will see in the final, we do not give dollar amounts, right? We're not going to tell you how much you should propose, or we're not going to give you, a ceiling. We expect you to price your solution accurately, and we'll evaluate based on, the entirety of your proposal. Solution summaries are mandatory. You cannot submit a proposal if you have not submitted a solution summary. So anyone who submits a solution summary is eligible to submit a proposal, but that first step is required. The big letters at the bottom of the slide. So those of you who have heard my talk before know that I'm going to say this as many times as I can while I'm up here. Read the ISO, right? If you have proposed to another ARPA-H program at any point in the past, do not assume that this ISO is the same as that ISO. Read it. You will notice this ISO looks different. We're trying out a different structure, but the contents might not be the same. So read the ISO and then read it again. Because the last thing you want is to spend time and money putting together a submission and have it rejected for something as simple as not following the instructions. So read the ISO. All right, other transactions. So other transactions are agreements. They are, in the legal sense, contracts, but they do not follow. We are not bound by the federal acquisition regulations. We are not bound by the financial assistance or grants and cooperative agreement regulations, right? So while they are bound by law, we have a lot of flexibility in what goes into those other transactions. When we post the final ISO, you will see I will be posting an other transaction for Delphi. It will show you kind of our going in position on the terms and conditions. We expect you to look at that. We expect you to ask questions. We expect you to provide us with red lines. That will be our basis for negotiation. Almost everything in that document will be on the table for negotiation, right? So again, this is unlike those other types of government contracts where here are the clauses we have to use, we can't discuss them, right? We're going to try to develop agreements that mimic commercial contracts more closely, but this allows us to get to a place that benefits both parties a little bit better than some other types of contracts. So understand, like I said, most of what you're going to see in those terms and conditions are going to be on the table for negotiation. We also recognize that a lot of you are new to other transactions. We're not going to throw you to the wolves, right? We're here to help. Again, ask all of the questions, especially once you see those terms. We will help you through it. We'll make sure you understand what you're agreeing to. All right, eligibility. So almost anyone who can satisfy our requirements can propose and be performers. Government entities are not eligible to be on performer teams in any capacity, not as a prime, not as a sub. So if you are considering teaming with a government entity, reconsider, right? FFRDCs, government agencies cannot propose. One of the questions we get a lot is about UARCs. UARCs are eligible to propose. So it is just really those government agencies and FFRDCs. SAM.gov registration is required for that entity that we are signing the contract with. You must have an active SAM registration when you submit your proposal. So if you are not in SAM, that's what I think you should plan to do tonight, is register for SAM. SAM can take a while, especially if you are a new registrant. There are a lot of very specific requirements that SAM has that you could miss very easily, and it could lengthen the time it takes for your registration to be approved and be viewed as active. So start that process as early as possible. We are not allowed to award to anyone who is not registered and active in SAM. Active means if you already have a SAM registration, check to see when it expires. You might need to spend some time tonight renewing your SAM registration because it needs to be active when you submit. It needs to be active when we make the award. It needs to remain active during the life of the program. Entities outside the United States are eligible to propose as primes or subs. Our statute requires us to prioritize work that is being performed domestically, but international or non-US entities are eligible to be a part of performing teams. I will say the caveat there is we are not allowed to provide funding to countries of concern. So there is that slight caveat. And of course, we can't make any awards, prime or sub, to anyone who is on a suspended or debarred list. And that includes if it's an individual on that list, not just an entity. All right, so the process. So I mentioned solution summaries are mandatory. When you see the final ISO posted, and as a side note, we are planning to post that as early as possible next week. There will be at Appendix B a template for you all to use for your solution summaries. These will be reviewed. They are not evaluated, right? And the feedback that you get will either be we encourage you to submit a proposal or we discourage you from submitting a proposal. You can submit a proposal either way. However, I would caution, right? If you receive a discourage, what we are saying to you is it is highly unlikely that a proposal will be successful. So again, it is up to you, but consider how you want to spend your bid and proposal dollars. Once solution summaries have been reviewed and you all receive feedback, then we move into the proposal stage. There is guidance at Appendix C that you will see posted with the ISO, and we provide templates for everything that you'll need to submit at the proposal stage. There's 4 evaluation criteria that are listed in the ISO. So all of the proposals will be evaluated against those criteria. We do not compare proposals to each other. We evaluate everything against those eval criteria. Those 4 criteria are extremely important because that is the sole basis for the evaluation, right? So make sure you familiarize yourself not just with the technical requirements, but with that eval criteria, because that is the lens through which all of your proposals are going to be viewed. This is the eval criteria that you'll see in the ISO. Again, we're going to release that as early as possible next week, so you'll have all of the details in writing, but these are listed in descending order of importance. Again, this is how we are going to evaluate your proposal. I'll wait for a moment. I see lots of cameras, so I will give you guys a moment. Right, this is the process. So with solution summaries, there will be a cover page, which does not count against your page count. Again, we provide you an Appendix B with the template, and we will ask for a rough order of magnitude of your budget. This is a very high level. We don't want to break down. We're not asking for a spreadsheet. It's at a very high level. What are your total labor costs going to be? What are your total subcontractor costs going to look like, that level? And you are not bound by that number, okay? We don't expect there to be wild fluctuations between your solution summaries and your proposals, but you are not tied to that dollar amount that's in your rough order of magnitude. At the proposal stage, you can see there are a lot of documents you'll submit. So you'll submit your technical and management proposal, the task description document. So for those of you who are familiar with government contracts, right, that's kind of akin to your statement of work on those contracts. The price proposal, this is going to be a narrative that accompanies the price proposal spreadsheet. Do not use that narrative to repeat what's in your spreadsheet, right? That does me no good. You use that to explain what's in your spreadsheet. Why are you pricing things the way that you're pricing? Where did you get these numbers from? Now, one of the things you're going to see if you've submitted to an ARPA-H program before, you're going to see that our price proposal spreadsheet is going to look a little bit different. We're not going to ask for the same amount of detail that you typically get asked for, right? We want to match our ask to the way that we're going to structure your awards. The awards will be milestone-based payments, right? So we're going to try to structure the pricing ask to more closely mimic that. So you're not going to have to, I mean, you still need to obviously internally build up your pricing in a way that makes sense, but you're not going to have to show us that same level of detail here. And then Appendix D is an administrative and policy requirements document. This is where you're going to talk to us about, do you have any entities or key personnel on your team that are outside the US? Are you planning to conduct human and animal subjects research? We ask about various items of, biosecurity and other such things. You'll need to provide for your key personnel biosketches. And there's guidance for all of this built into that document. Once we receive your proposals, they'll be evaluated through our scientific review process. You will get a notification saying you've either been selected to move forward for negotiations or you have not. If you have been selected, we are going to handle negotiations a bit differently. You notice there's a trend here. We're doing a lot of things different on this program. I appreciate Paul letting me just have freedom to play around with his program. So historically, if you've negotiated with ARPA-H before, there's a lot of back and forth over email. Sometimes we'll get on the phone. What we're going to do here is we are going to schedule a time to negotiate. We are going to negotiate everything with you during that time, right? Your technical tasks, your price, the terms and conditions. So what that means for you is you need to come to that meeting with all of your decision makers at the table, right? There isn't going to be we'll take this back and no, your decision makers need to be at that table. We intend to walk away with a full agreement between the two parties so that we can move to awards and funding much quicker than we normally do. So plan for that if you receive a select notice. All right, teaming is likely going to be necessary for Delphi. You do have to submit a proposal that demonstrates that you can accomplish all of the technical areas. Do you have to team with other entities? No. If you think you can do it all in-house, great. But it is highly likely you all are going to need to team. Please take advantage of today. This is a wonderful opportunity to build your team. Those poster sessions, even if you didn't bring a poster, you should be in there looking at posters and talking to people. We also have the Delphi teaming site, so you can, go on there, sign up, list your capabilities, and use that as a way to find others who might have capabilities that your current team is missing. All right, so we are going to live by the ISO. Anything that is said today is trumped by the ISO. We do try to make sure everything is consistent, but just in case, I want to say that. So again, make sure you read the ISO, read all of the attachments to the ISO. If we post an amendment to the ISO, read it, because it will override something that was in the original document. Questions should be submitted through the Ask a Question site, with the exception of today. If you're online, submit to the Delphi program email. We do post the questions and answers publicly on the program page. Make sure, again, that you register in sam.gov and our submission portal as early as possible, so that if you have problems, you can get help before the deadline. The other thing I will emphasize, and Ed mentioned it earlier, if you are going to sign up for a one-on-one session with us today, don't use that to pitch us your idea. It's not going to make any difference. You will not get an award because of any conversations that we have today. Use that, again, find out if what you're talking about is in scope, ask other questions that you might think of between now and this afternoon, but it's not a time to pitch, right? You are pitching in your solution summaries. And also read the ISO and all of the attachments to the ISO, and then read it again. Right. I will be around all day, so if you have questions, feel free to grab me.
I just wanted to make one comment. Well, one, thanks to Adria for a great presentation. Really appreciate it. And also for the new director. It's all about going blazingly fast. And that's part of these innovations that she's bringing forward. Sit down, because that back and forth, that negotiation often takes forever. So we're going to sit down and we're going to get it done fast. And the intent, we're not giving a date, but the intent is to get this done very, very quickly. Yeah, if you're thinking about what fiscal year thing, you can think about getting underway by August 1st.